Hello everybody, my name is Dr. Zahid Mahmood and I am working as Principal Scientific Officer at Wheat Program, Crop Sciences Institute, National Agricultural Research Center, Islamabad, Pakistan. Today I will talk on rapid utilization of wheat genetic resources implying speed breeding, machine learning and AI-driven techniques. First of all, I will show you the world wheat production scenario. Pakistan lies between the 20 to 40 million tons per hectare production per year, and last year we harvested 31 million tons in Pakistan. We are facing major challenges in wheat productions like biotic stresses, abiotic stresses, crop management, and biofortification. In biotic stresses, diseases especially yellow rust, stripe rust, and stem rust, then weeds, insects, and pests. In abiotic stresses, terminal heat stress, temperature fluctuations, severe drought and erratic rains, and soil salinity. In crop management, we are facing late planting, use of non-certified seeds, inadequate and imbalanced use of inputs, poor management practices, and again we have malnutrition problems, and so we are working on the iron and zinc fortified wheat varieties. As I earlier mentioned that major diseases like rust, they are very important, so we choose 400 wheat genotypes including synthetic hexaploid and synthetic derivatives. Synthetics are produced by crossing Ageloptoceae to the Triticum turgidum, and synthetic derivatives developed by crossing synthetics to adopted weeds. These holds great variation in agronomic traits for biotic and abiotic stresses. And then we did screening of germplasm against different races of stripe rust, stem rust, and leaf rust. We evaluated at different locations in Pakistan, Australia, at seedling and at the adult plant stage. We use these following rust pathotypes in Pakistan and Australia. After evaluation of the germplasm against different rusts like leaf rust, stripe rust, and stem rust, we decided rapid development of rust-resistant wheat using speed breeding technology. Wheat breeding technology adaptation in Pakistan. Generation advancement time is a major bottleneck for plant breeding in Pakistan. So conventional wheat breeding takes 12 to 15 years to develop and register a new wheat variety in Pakistan. So we adopted shuttle breeding in 70s, and we are taking 2 generations per year. But again, when we plant second generation, it's a very rainy season, so in the field plants are not much healthy. So recent times we adopted speed breeding for rapid generation advancement. So what is speed breeding? It's a NASA-inspired set of techniques that involves the manipulation of environmental conditions under aiming to accelerate flowering and seed set to advance the next breeding generation as quickly as possible. Evolution of speed breeding. NASA started in 1980s to grow plants in the space station, and then in 2000 University of Queensland started working on speed breeding techniques, and ultimately in 2018 with the collaboration of John Innes Institute and University of Queensland, they published first speed breeding protocols in Nature Protocols, and in 2021 Pakistan established its first speed breeding facility for wheat generation advancement, and now we have 2 facilities, one for wheat and other for pulses crops. Why speed breeding? Where speed breeding fits? It fits off-season, rapid generation turnover. Then we can have a rapid hybridization. We can bring the breeding program indoor, a rapid screening for different traits like heat, drought, and salt tolerance. And speed breeding can be integrated with genomic selection as well. So it's a very flexible sort of technique, and speed breeding has the potential to reduce the time required for cultivar development and registration. This is Pakistan's first speed breeding facility to fast-track varietal development, which we develop under the Wheat Productivity Enhancement Project funded by PSDP Pakistan. What is the difference between traditional and the speed breeding pipeline? Basically, we are putting a lot of time, 8 years in crossing and inbreeding time. So in speed breeding, we just address this problem 8 years. We reduce it 1 to 2 years, and we can have a rapid speed breeding pipeline for development and registration of wheat varieties. So seed to seed speed breeding system. In this system, we maintain the temperature 17 to 22 degrees centigrade in the glass house. We maintain humidity 50 to 70%, and then we provide 22 hours of photo period, and we keep in mind to provide ppfds 450 to 500 micromole per meter square per second. Then scaling up, because every light have its own potential of production of light spectrum, so we maintain these ppfds. And then we harvest little earlier, because when the spikes are turning yellow after 15 days of anthesis and seed is hard, so we cut and we dry it in the hot air drying oven at 35 degrees centigrade. So in this way, this system takes 6 to 8 weeks from seed to seed, and we can harvest generation just in 6 to 8 weeks, and we can harvest 5 to 6 wheat generations per year. So these are the key areas of the spectrum for plants, like the blue spectrum, then the red is very important, then the far red is very important. So we are using LED grow lights, heliospectra LED grow lights, and these lights are very flexible and controllable, and we can program these lights according to our requirements. And in this way, by providing the specific temperature, humidity, and prolonged photo period of 22 hours, actually we created the plant factories. In these plant factories, we can manage a lot of number of plants inside a small place. So even we can harvest good seed from, if we are planting 1000 plants per meter square of wheat and barley. So again, if we are using hydroponic technique inside the speed breeding facility, we can have more healthy plants. So now I will come to the results of rust resistance and association genetics. We identified 30 yellow rust resistant synthetic derivatives, 18 resistant to all 5 races and also at both locations. 31 MTAs and 27 MTAs found associated to yellow rust resistance. Identified 10 synthetics having triple rust resistance, 24 having APR for triple rust, and 57 having LR, 77 having YR, and 69 having SR resistance. The diagnostic competitive allele specific markers, CASP markers, revealed that 14 synthetics had LR34, 80 carrying LR46, and 3 synthetics carries LR67 genes. 41 MTAs found associated with leaf rust, yellow rust, and stem rust at seedling and adult plant stages. loci on chromosome 1D, chromosome 3D, chromosome 5D were associated with yellow rust and stem rust resistance, and chromosome 7D with leaf rust, yellow rust, and stem rust, likely to have novel alleles for rust resistance in synthetics. Studies showed great potential of synthetic derivatives, synthetic hexaploid weeds, germplasm resistant against rust diseases. Speed breeding result. We are advancing generations in 56 to 64 days. We are harvesting 5 to 6 generations, and we maximum harvested 31 spikes from one F1 plant and 88 seeds from this plant. Means we can have healthy plants by doing speed breeding. We performed rapid screening analysis like heat and drought and some root phenotyping, and these are very rapid because we can perform in 2 months. We developed 265 population, developed and advanced through speed breeding facility just in 2 years, and we developed 4,000 wheat lines through speed breeding, and now you can see these lines are under the field trials. And we are also providing trainings to national and international scientists. So in this way, speed breeding can save 6 to 7 years. Now you can see here the wheat population developed through speed breeding under the field trials. After having the production and developing of a large number of plant populations, we decided to combine speed breeding with high throughput phenotyping for rapid breeding pipeline, because when you have a large number of plant populations, it's very difficult to phenotype them in the field. Because the phenotype of large number of population in the field is a major bottleneck, so we decided to use UAV based platforms for high throughput phenotyping. People are using different the platforms for phenotyping, and these can assist in early selection in the field through NDVI, canopy temperature, pigment senescence rate, stay green, plant height, spike number, ground cover, and yield prediction. So ultimately our goal is yield prediction, how we can easily predict the yield of a large number of plots. It can cover large experimental plots at multiple time and locations. It can accelerate the accuracy for genetic analysis. So we are using different UAV based aerial phenotyping platform. You can see here different drones we are employing for this phenotyping. We are using different the multispectral and thermal sensors. We are using MicaSense Ultim PT, MicaSense Red Edge, Parrot Sky, Centira, and DJI Multispectral M3. Speed breeding population at NARC. Now you can see this is a recent picture of my breeding population plot, and in this plot we have about 6,000 wheat genotypes. So for this plot we recorded NDVI, canopy temperature, early ground cover, plant height, yield, and ultimately we predicted yield for different plots. And not only for wheat, we also did high throughput phenotyping using multispectral sensors and thermal sensors at NARC. So until now we phenotyped 4,000 wheat plots, 1,100 potato plots, 1,300 pulses genotypes, and 500 rice genotypes, and 1,200 different oilseed crop genotypes. At the end, I will tell you we are also employing the machine learning, deep learning, artificial intelligence, and IoT for the sake of the rust evaluation and categorization. We are using AI-driven disease detection classification technique. We develop a prototype which can detect the rust and which can classify the rust into, for example, in resistance, moderately resistant, and susceptible genotypes. We are also employing IoT UAV based crop health monitoring. We are using different IoTs, and we in the field, and we can have the live monitoring of crop health in the field. It can cover the large experimental plots at multiple time and locations. So speed breeding helps in rapid generation advancement, and high throughput phenotyping accelerate the accuracy for genetic analysis. So in this way, we have a large number of the genetic resources conserved in the gene bank, and we can use them, and we can rapidly integrate the new genes, incorporate the new genes in our new populations, and we can accurately phenotype them in the field in a short time. And we are having national and international collaborations with different institutes, universities, and research institutes. Thank you very much, and if you have some questions, you are welcome.